So I think it's time to get a little bit more revolutionary. So please extend your warm welcome to our next guest, Professor John Bickle from Mississippi State University, who will tell us about what kind of scientific revolution was the discovery of mammalian adult neurogenesis. So thank you so much for coming, please welcome Professor Bickle warmly.
Great. Thanks so much. Thanks so much. First of all, just a thanks to the entire Open Lab who put on this wonderful summer school and this great ancillary session. I've been just enjoying myself. My first trip to Poland ever, so it's been wonderful. So the title of my talk may puzzle some of you, because some of you might be thinking, isn't there only one kind of scientific revolution? Every philosopher of science knows it. Many scientists know it. We teach it to our undergraduates, and especially when we're introducing them to the methodology of sciences in our textbooks. And its entire jargon is now across the academy. It is, of course, Thomas Kuhn's paradigm shift account of scientific revolutions that Kuhn first introduced in his 1962 book, and it's come to be developed on. And it's now the words of anomaly and crisis and paradigm are, of course, part of everyday academic discourse. Now, in summer of 2022, I started a new research project with a new collaborator, David Parker, who's a neurophysiologist at Cambridge University in the UK. And our first result is a paper that just came out earlier this year, a chapter, Revolutions in Wet Lab or Wet Neurobiology, which came out in a SAGE volume on cognitive and systems neuroscience. And in that particular chapter, we talk about 3 case studies. The late 20th, early 21st century discovery of adult mammalian neurogenesis, the late 20th, early 21st century discovery of long-term depression as a complementary process to long-term potentiation, and the development over the last 100+ years of the microelectrode and its extensive and continuing uses in neurophysiology. And the lesson that we stress in this particular chapter is that not one of these revolutions is Kuhnian in structure. And that's what I want to talk about today. Okay, so now in this particular chapter with regard to adult mammalian neurogenesis, we insist that one key component of Kuhn's picture of revolutions in science is missing from this particular episode. And that is the fact that a Kuhnian paradigm must function to guide ongoing normal science. And that key feature is simply not there if you have a no new neurons approach to neurogenesis. No new neurons offers no specific guidance for pursuing further research, for filling in gaps in our knowledge, for expected further findings. No new neurons is not a Kuhnian paradigm in this one non-negotiable sense. Instead, what we argue is, when you look at this episode of adult neurogenesis, this is an example in which a pre-paradigmatic science moves into its first paradigm, that being that adult mammalian neurogenesis is real, which now lays out an entire set of experiments for you to perform. You can figure out where it occurs in the brain. You can try to figure out what its mechanisms are. You can try to figure out if you can use those mechanisms for clinical purposes. So what happened, we argue in this chapter, when adult mammalian neurogenesis was discovered, was that part of developmental neuroscience entered into a new phase, a genuine paradigmatic phase. Now, I'll admit that the idea for going this direction was David's. We engaged in a number of exchanges, and he's the one, because he had actually published a previous paper where he argued that the discovery of adult mammalian neurogenesis was Kuhnian in structure, and I convinced him it wasn't. And so he came up with the idea of this move from pre-paradigm to paradigm stage. But I insisted at the end that we agree on a footnote. And that footnote says, a case can also be made for adult mammalian neurogenesis being a new research tool-driven revolution in the sense of Bickle 2016. And this is the paper I spent a lot of time talking about at the summer school. This 2016 paper is of mine, Revolution in Neuroscience, Tool Development, where you will find this quote, for revolutions in neurobiology, Kuhn's account is wrong. There have been revolutions and new ones are brewing, but they do not turn on competing paradigms, anomalies, or the like. Instead, they turn exclusively on the development of new experimental tools. That was the theme in that book. And I want to make the case out for that today, for the adult mammalian neurogenesis case from late 20th century neuroscience. Now, I'm not going to have time to talk about some scientific details of neurogenesis. The textbook definition of neurogenesis is the process by which new cells are formed in the nervous system from stem cells and progenitor cells. But what's really interesting in this regard, and I'll just mention this, is that both glial cells, for which nobody has ever thought that they didn't get generated in adult mammals, and new neurons actually come from the same set of neural stem cells. It's just a different feature of the progenitor cells that drive it, and most of the time they're driving it to new glial cells. But there's another aspect of adult neurogenesis, and that is the incorporation of those newly generated neurons into functional cortical circuits. So adult neurogenesis would be the process by which this happens postnatally in, quote, mature organisms. But as you're going to see, there's some dispute about what constitutes an adult in this particular case. Okay, so for most of the 20th century, the widely accepted view was no new neurons. And this goes back to the grandfather of neuroscience, Santiago Ramón y Cajal, the great Spanish neuroanatomist, who in a 1928 book gave an assertion that no new neurons were generated post-birth, and also gave what turned out to be a really compelling argument for this. Neurogenesis in an adult mammal would interfere with the necessary long-term stability of both inherited and learned cognitive processes realized in neural circuits. He gave that argument back in 1928, and that argument continued to convince really important neuroscientists up to the turn of the 21st century. But look, Ramón y Cajal was no dogmatist. After that famous passage, he closed with, "It is for the science of the future to change, if possible, this harsh decree." Okay. Well, in the early 60s, a new report of neurogenesis appeared in an issue of Science that came out on the 30th of March in 1962, single-authored by a young investigator at MIT, Joseph Altman, who was using a then new research tool, 3H or tritiated thymidine, as a radioactively labeled NDA precursor, the T in the GCTA series, detectable by X-ray autoradiography, and he was using this tool to investigate glial genesis in adult mammalian brains. But in this 62 Science paper, he published a remarkable finding. An unexpected finding, light microscopy evidence of labeled new neuroblasts and differentiated neurons in adult rat and cat hippocampus and olfactory bulbs. So he actually found that radioactive label that would have been a new neuron after the time that the 3H thymidine was injected into the animal and the time that the animal was sacked and looked for new cell genesis. There's one of his examples from the couple of examples. That's a labeled cell. In that case, that's a labeled hippocampus cell that he pointed out as an indication of a cell that had been generated because of that tritiated thymidine between the time that the thymidine was injected and the time that that particular slice was taken, and that was a new neuron. Okay, he followed this up 3 years later with a paper where he and his graduate student, Gopal Das, used the same tool, the thymidine, the autoradiography and light microscopy, and he reported evidence of more examples of newly generated neurons in an adult, in this case a rat. Here's a labeled granule cells in the dentate hippocampus gyrus. You can see all of those particular cells are labeled with the thymidine, the radioactive thymidine, at different magnifications. And the nice thing about this example, that's a layer of hippocampus at which you do not find glial cells. And so he was convinced that those cells had to be neurons because he was specifically looking at a region of hippocampus where the glial cells are not there. He also found, remarkably, a pool of undifferentiated cells along the lateral ventricles in various phases of mitotic activity. So it's as though there's a whole pool of new stem cells and other cells waiting to be differentiated into neurons. Then, in the late 60s and the early 70s, Altman, Das, his graduate student, and Shirley Bayer, a new postdoc in his lab, used the same labeling technique, but they also coupled it with use of low-level X-ray induced cell death and some behavioral measures. So what they did was they showed that by destroying those newly generated neurons in the hippocampus, the adult hippocampus of the animal had about 85% less granule cells in the hippocampus, which was very close to the amount they estimated that we would have on some of their earlier studies. And by doing the behavioral work, they showed by using a number of behavioral measures that these degranulated animals had the same pattern of responses to various memory tests that are sensitive to hippocampal damage. Okay, well, that looks like a convincing set of evidence over a 10-year period of research. How did the neuroscience community react to Altman's findings? Charles Gross, the famous Princeton neurophysiologist, in 2000 published a paper where he laid out some ideas of revolutions in neuroscience. And one of the cases he talks about is adult mammalian neurogenesis. And now I don't have time in this short of a talk to go through what Gross says about Altman's work in this paper. So I'll just tell you what Altman said in a review and kind of a look back article that Altman published in 2011. He says, Charles Gross leaves the impression in his historical review that because I was an unknown scientist, my early publications could be ignored by the neurobiological community. That's the kind of story that Gross suggests. Being a young scientist, being a relative unknown, working in a psychology department, it was perfectly understandable why the broader neuroscientific community just didn't pay attention to Altman's work. But in that 2011 recollection paper, Altman gives some really convincing evidence that a lot of people, when he first published this stuff, were taking it seriously. We got multiple invites, he got multiple editorship offers, he got grant funding up into the early 70s. He had in fact, he was on the list of the 1,000 most published scientists between the 60s and the late 70s. So, now, I'm not going to talk about this today, but I urge you, if you have some interest in the way that someone who has basically been bypassed in a scientific discovery, go take a look at this 20,000 recollection paper and account. It's a fascinating in and of itself, but I won't have time to talk about the way Altman remembers what happened to his career. Okay. let's thread forward to the late 1970s. Because now a new young researcher picks up on this idea of neurogenesis. But now in the late 70s, he's got a new research tool. He's not only got the 3-H-thymidine, he's not only got all the machinery to do that, but he's got electron microscopy to look at these cells under. So he's cutting much, much thinner slices to do the electron microscopy. So you don't have to worry about a glial cell just being superimposed over a neuron, which you have to worry about when you're cutting thicker slices through hippocampus and otherwise. And we can now look at the microstructure of these particular cells that are labeled with the thymidine. And he finds newly formed cells with neuron ultrastructure in the same brain regions where Altman has found them, dentate gyrus, olfactory bulb, lining the third ventricle, plus he finds some in visual cortex. Let me just show you one example of this, because what he publishes in this science paper are, and you see on the left here are light microscopy images, and on the right are the electron microscopy images. And if you look, especially my favorite is C and D down there. If you look at D, the electron microscopy image of that particular, that's a neuron. Those are postsynaptic densities you're seeing around there. Those are vesicles of neurotransmitter. That is not a glial cell. However, Pasco Rakic, even at that time, probably the world's most prominent developmental neurobiologist, uses that same tool, the 3-H-thymidine and electron microscopy in his primates, and he finds absolutely no evidence of new neurons, new neurogenesis. Every single thing in his that gets labeled is something that is prenatally or immediately postnatally. And that takes the wind out of the sails when Pasco Rakic can't find what other people are reporting. And it's really interesting. At a 1984 international conference in San Francisco, where both Kaplan and Rakic presented their latest data, as did Shirley Bayer out of Altman's lab, who by that time was Altman's wife. And Rakic remarks in a public discussion to Kaplan, those may look like neurons in New Mexico, but they don't in New Haven. Kaplan ends up leaving research science after this meeting. He goes and practices medicine for the rest of his life. A year later, in Science, Rakic publishes his data of no new neurons, and the study of adult neurogenesis in mammals is virtually abandoned. Well, here comes the early 80s, and now we get a cool new animal preparation. Fernando Nottlebaum at Rockefeller University in New York starts actively looking for adult neurogenesis in songbirds, canaries, whose telencephalic song production regions actually change in volume over the seasonal and increase in size during the mating behavior season. And he finds new neurons with this 3-H-thymidine and with the microscopic structural analysis. And even more interestingly, he and his postdoc, John Patton, use a brand new research tool, the patch clamp, the whole cell recording patch clamp, and painstakingly show that some of those newly labeled neurons that were generated in the adult phase in these songbirds actually incorporate into sound responding circuitry. What was the reaction from Rakic and the other heavyweight developmental neuroscientists about Nottlebaum's findings? Well, Fernando's found a cute fact about bird brains. Rakic and his colleagues throughout the 90s in every major neuroscience journal argue no new neurons in mammalian brains, adult brains. And they stress the very same argument that Ramon E. Cajal had given, back in the late 20s. It's just that would interfere with the necessary stability to maintain the necessary social and cognitive factors in mammalian behavior. Well, let's stop mid-revolution here for a second and pause and think about what's going on here. This tool, 3-H-thymidine was brand new to biology. Its first use was in this 1957 Proceedings of the National Academy of Sciences paper, Taylor's, that came out of the Brookhaven National Laboratories. This was introduced, the 3-H-thymidine was introduced to replace radio-labeled carbon in search of, quote, the macromolecular structure of chromosomes and their mode of duplication. And remember, we're here in 1957, only 4 years post-Watson and Crick. So, what they do is, the reason why they chose this particular tritium, this 3H label of thymidine, was because it has a very short range of beta particles. And so it can therefore make identification of labeling possible in particles as small as individual chromosomes. And the thymidine, of course, was used because that would be the particular, that's going to be the piece of the new DNA synthesis as a unit. And they were able to show some remarkable facts using this label. And what they did was they showed that these chromosomes, the length of the chromosome, which remains intact throughout the succeeding replications and nuclear divisions. Hugely important findings just 4 years post Watson and Crick. A year later, the first use of this particular tool in mammals is reported out of Brown University. First reported use of 3-H-thymidine in mammals, mice in this case, to empirically demonstrate greater increased amounts of cellular proliferation than had previously been estimated, a longer period of DNA synthesis, and differing rates of cellular integration in differing biological tissues. Again, remarkably important findings at that time. So this tool was brand new. When Altman started using it in the early 60s to explore glial genesis and found neurogenesis, it had only been around for a few years. So it meets all of my 2016 meta-scientific derived account of developments of revolutionary tools in neuroscience. The motivating problem for this particular tool: the need for a label detectable by existing technology, X-rays, with short enough beta particle radiation range to identify the radio label, the tritium, in individual chromosomes, and the need to be attachable to a component specific to newly synthesized DNA and detectable across numerous replications and nuclear divisions, thymidine. That's what they needed to do this early genetic research. First phase hook experiments, the two I told you about, the 57 Taylor paper and the Hughes 58 paper. Here's one of my conditions on a first phase hook experiment, an initial phase hook experiment, published in a major venue, PNAS. Using targeted phenomena central to the specific field for which the tool's development, early molecular genetic, a common plant preparation that Taylor used, and a common rodent preparation that Hughes used. Generating groundbreaking results, well, you saw them: DNA synthesis, tracked through replication cycles, empirical corrections to previously quantitative estimates, hugely important findings in the field of early genetics, early structural genetics. So Altman's and later Kaplan's work would count as second phase hook experiments in my model, by which a new research tool and its results is brought to the attention of a wider scope of scientists besides those just in the field for which the tool was developed. Okay, one more new tool that was central to these results. I mentioned Nottlebaum and Patton's work, the patch clamp. The patch clamp, when Nottlebaum and Patton began using it, that 84 publication had only been in existence for about 4 years. It was first developed with a paper in Nature in 1976 by Naher and Sackman, But it was another 4 years until Naher and Sackman worked out the details of how to achieve the gigaseal between the glass micropipette and the polished self-surface. It was another 4 years before they worked out how to achieve the gigaseal, and I tell that whole history in my Tinkering in the Lab paper, which I sent in to the school for reading. Okay, so now something new happens at this point. Rakich and crew are still carrying the weight. No new neurons. But then, all of a sudden, the adult neurogenesis revolution resumes with new evidence emerging in the 1990s. Elizabeth Gould, who did her work in Bruce McEwen's lab at Rockefeller, then at her own lab in Princeton, and of course one of the superstars, Fred Gage, his lab at the Salk Institute in Southern California, produced new evidence that adult mammalian neurogenesis is real. And what they use is a new tool. BRDU, a thymidine analog, BRDU, which incorporates into newly formed cells. It is detectable by immunocytochemistry, a brand new tool. A brand new tool where you use antibodies to indicate the existence of a particular protein. In this case, one that is sensitive, that indicates the existence of the BDRU incorporated into the newly synthesized DNA, plus other antibodies which can be specific to the kind of tissue that you're examining. Gould quickly replicates with this new tool Altman's and Kaplan's earlier findings in a variety of mammals and extends them to primates, neocortex, and Gage's lab basically finds neurogenesis in adult human hippocampus. In cancer patients at autopsy who underwent BRDU treatment while they were still alive clinically, to visualize tumor growth. And then after they died, you could look in their hippocampus and find indeed new, in their olfactory bulbs, newly generated neurons in these elderly, about to die cancer patients in human hippocampus. This new evidence is fiercely disputed. And if you go back and look at it's one of the most beautiful publications of scientists opposed to each other about a technique. One of the best publications is this commentary, this technical commentary in May of 2000, where Gould and Gross are arguing for, and a couple of other people are arguing against, and it's just a beautiful step-by-step process of explaining how this particular procedure with BRD uses, and one side challenging it, the other side defending it. But more evidence continues to arise, and by the early 2000s, even Rakich finds new neural genesis in his adult primate monkeys and reports it, especially with the BRDU technique. Another decade and a half of confirming evidence occurs, and the adult neurogenesis revolution is widely accepted across all of neuroscience. Now let me just show you that this still presents. So let me say, so the new research tools are at work here again. BRDU, that particular labeling technique that became so important in the 90s, using immunocytochemistry instead of autoradiography, that had actually been in existence for about 10 years. So it was not totally new, but new in a neuron-specific nuclear protein in vertebrates was only first discovered by Mullen and collaborators in '92, who were investigating a series of mouse-specific microbial antibodies for use in transplantation experiments. So basically, the indicator that those particular new cells were neurons, that had just recently been discovered when Gould and Gage's lab first put it to use. And of course, it drove the new evidence that ultimately became convincing to enough people. Now look, the new received view of the circa 2018 Okay, you're here looking at a pair of papers, the top one published in Nature, 3 weeks later the second one published in stem cell, top journals in the field that have virtually contradictory conclusions. Human hippocampal neurogenesis drops sharply in children to undetectable levels in adults, and human hippocampal neurogenesis persists through aging. A group of people 3 months later did an extensive investigation of the methods used in these two papers. Notice two of the authors on this paper: Human Adult Neurogenesis, Evidence and Remaining Questions. Fred Gage was the second author, Elizabeth Gould was one of the later authors, and after this thorough compare and contrast between the methods used in these two papers, they conclude, "We argue that there is currently no reason to abandon the idea that adult-generated neurons make important functional contributions to neuroplasticity and cognition across the human lifespan." That's what this group of 17 researchers put their names to. Adult neurogenesis is real, and it persists through the life cycle. So, yeah, the revolutionaries, now the establishment. Okay, so to appropriate a Hindu mythology that someone else appropriated yesterday at the school, it's turtles all the way down. When it comes to revolutions in neurobiology, it's new research tools development and their use all the way back. That's the number of papers I've been publishing on a bunch of these different kind of tools to try to make out the case that that is the driver of new scientific discoveries and scientific progress, not anything like Kuhnian paradigms and anomalies and the like. One finds cases for this in the development of the discoveries initiated by the metal microelectrode. I talked about that in a couple of papers. The patch clamp, I've gotten a whole history of the development of that and its impact. Gene targeting, which I talked about in the school the other day, and optogenetics and relatively more popular now, chemogenetics. All of these follow the same trajectory with regard to the development of this tool and the impact of this tool in making the new discoveries that lead to progress in neurobiology. I'll close with a quote from Sidney Bruner that my friend Anne-Sophie Barwitz sent to me when I was in the middle of writing all these papers on tools. Bruner, of course, was the guy who won the Nobel Prize in 2002 for bringing C. elegans into neuroscientific investigations. And he is quoted as having said, "Progress in science depends on new techniques, new discoveries, and new ideas." Probably in that order. Yay. Tools first. I welcome any of your questions and discussions because I think I've now worked out this kind of approach for this other important revolution in recent neuroscience, the discovery of adult mammalian neurogenesis, and quite literally, it is tools that drive neuroscience progress, not theories. Thanks.
Okay. Thank you so much for this talk.
Sure.
So now we are open to questions. Does anyone have a question?
Great.
Okay.
Great. Awesome.
Thank you very much for the great talk. Many of us have been witnessing this debate for years, and but the way you presented it, it kind of highlights the one specific feature of this debate, and that actually answers the questions that we had before to Danielle. What is the difference between artificial intelligence and human intelligence? I don't think artificial intelligence would really stick to its beliefs for that long just because of its ambition and the prizes and being the first to publish and not being the last to publish and so on. I think artificial intelligence, if there were two of them from two different makers, let's say, or two different sources, they'd agree much faster than
than human beings.
Two opposing camps in this debate. So that strikes me. I mean, it strikes me that it took so long just because of human ambition and these little petty.
Yes.
I don't know, animosity or whatever.
Yeah. And the very institution of science, the very institution of the way science is institutionalized, on those kind of measures. How do you measure your productivity? I mean, people ask me, I point to my H index and my, my I-10 index and grant funding, and that's how we get ahead in science.
Or maybe if you put artificial intelligence into the same system, it'll behave the exact same way. So maybe
we'll see. We'll just have to wait and see. We don't know.
Okay. Any other questions?
Thank you for the talk. I just wanted to ask, it was pointed a couple of times about the functional relevance of those neurons that are developed in adults. And I don't know, I don't think there was any evidence presented, and I don't know whether it was lack of time, but I would like to ask if there actually is any evidence apart from the very molecular level of evidence that those are actually incorporated into the small neural motifs and something bigger like actual cognitive effectiveness or something. Yeah. Thank you.
No, that's an excellent question, and the answer to that question right now is, I mean, when people still go back and cite that Payton and Nadelbaum paper to show about the incorporation, but that's not mammals, okay? And so now, now Paul Franklin, for example, is one of the authors of that 17 or 18 author paper. Paul Franklin's been doing a lot of work lately in his lab that he shares with Sheena Jocelyn, his wife, on showing that these particular new neurons do indeed get insinuated into functional circuitry. But that's really hard work to do. And again, Paul's work has also been criticized on strong grounds that he's not actually showing that. But the thing is, Nadelbaum and Payton did perfect a method that's still used today, to where you can patch on to those particular cells, and you can patch on to a bunch of cells in the region in which the suspected neurogenesis has occurred, and you can record from any cell that you can find that's responding to sound, and if you're in that circuit, and it's responding to the stimulus, you can record from those, and then you can go back and see how many of those cells that you recorded from when you were doing your patch clamping in the intact animal, after you sack the animal, whether or not, how many of those cells are labeled for whatever you're using to label newly generated neurons. It's tricky work, but it is being done, and there is evidence that these new neurons in hippocampus and in olfactory bulb in particular are insinuating into functional circuitry. But again, it's tricky work. There's not only is it difficult to do, you're still having to convince people that the very same cell that you're doing your patch clamp recording from was actually one of the labeled cells. So it takes intense histology. It's just really hard research. But there is some evidence using this technique that was developed back in 1984 that suggests that, and just updating it with immunohistochemistry and stuff like that, to suggest that yes, these are insinuating their cells into functional circuitry. But great question.
So I'm going to raise here to in defense of Paul's work, and because it does make perfect sense. I mean, there is a feature of this paradigm that allows easy incorporation of the adult neurogenesis, right? So our memories are kind of fluent, and they're bound by events that happen together, right?
Yes,
so if you incorporate that, you might think there is a need, there's an actual need, not only it allows incorporating these neurons, there's actual need for having these neurons. It's if you think out of the box, you think you discover that actually you would want them to be there because you experience new new stuff every day, new events, and then we want to bind them together. But we want to distinguish between the periods of our life that are that are separated by longer periods of time. So these events, or these molecular events and psychological events, let's say, they happen on a very similar time scale. So the presence of these neurons allows us to create memories that are like a chain of events. So for example, today's symposium will be bound together with the upcoming weekend in your brain, but not necessarily with another symposium or another weekend. So on the level of physiology, biochemistry, I don't know, neuroplasticity, this all makes perfect sense. It does not, it does not really it doesn't really counter Ramon and Cajal's observation about the stability. And if you think about even the work on patient HM, his retrograde amnesia was not was only limited to certain events several years back. But so these were the ones that were affected with the hippocampal but then some older events were still in his brain. So another idea is that these hippocampal events don't stay there. They've been transferred to another areas of the brain. So there's the idea of systems consolidation. And that also makes perfect sense and perfect room for these newborn neurons. So there's actually absolutely no discrepancy between our general idea of how brain works and this new discovery. So
especially now that we've got more of the molecular biological story as to how these memory linkings take place, right? To where now we know about the neurons that are activated around the same time do tend to get linked. And so if there are components of a memory, that's another way of incorporating one of these neurons directly, but you still got to physically grow the connectivities.
I know that Paul's results at this point, they look very clumsy because this is really challenging technically.
Yes, as I mentioned, it is. This is hard work
to follow them. And he's aware of his let's say setbacks. But I think it's going in the right direction. There's actually no discrepancy between theoretical ideas and their experimental work.
I couldn't agree with you more. And I think that the more that basic molecular biological biochemistry story is being worked out about how memories get linked via the way these neural activities get linked. Paul is beginning to incorporate all that into his work, and I think it's on the right track for showing that yes. And your point you made as well is that, look, memories are not as stable as our sort of folk psychology kind of makes them out to be. And we've known that from cognitive science. I mean, just cognitive psychology for, Endel Tulving was pushing that back in the 80s. I mean, so I think you're right on.
And I have just two, I think, small questions.
Sure.
One is, I was wondering what you think, how far you would stretch the term tool. I mean, I was wondering whether you would count the computational tools.
Good.
I mean, as modeling tools, as tools, or maybe as a theoretical framework, which is distinct from tools.
That's an excellent question.
And a second one, which is, I think, connected. Would you think that if tools, maybe distinct from theories, drive progress in neuroscience, is it really good? A normative question.
Oh, good. No, on the first one. So, the computational tools. Look, because of my scientific interest, which is wet lab neurobiology, I've concentrated on the tools that have been important in wet lab neurobiology for the last 30 or 40 years, primarily. Because that's where my own interest is. Do I think that a similar story can be told for the development of other tools? Well, computational tools are a little harder, right? Because there is sort of because the story I tell about tool development, I didn't tell about it here. The story I tell about tool development is that tool development generally comes about from this kind of atheoretical tinkering in the lab. There's a theory in the background for the motivating problem for the tool, no doubt about that. But the way you build those tools is very much laboratory tinkering, just make the damn thing work. So that's my picture that theory is actually tertiary in importance in wet lab neurobiology. It depends on new research tools, and the new research tools depend upon this kind of atheoretical laboratory tinkering. I don't think that similar story can be told for computational tools. In fact, I think that's where I think we're seeing a fundamental difference in the scientific practices involved in kind of the wet lab neurobiology that involves cellular, molecular, and behavioral neuroscience and computational neuroscience. I think one of the reasons we're not finding those to be so easily integrated is because computational neuroscience is quite literally a theory-driven science. It's kind of like physics in that regard. And wet lab neurobiology is an experimental tool-driven science where the tools are very much the result of a process we might as well call tinkering. So I'm never surprised when we fail to find integration between these two levels of science. And that used to bother me, but it doesn't anymore. Why does neuroscience have to be unified in the way it approaches problems? The wet lab neurobiologists can do their things, the computational neuroscience can do their things. Why do we have to meet up somewhere? I mean, we'll just keep telling our story. We got fundamentally different pictures about what the brain is. The brain's a chemical stew, or I'm from the South, a chemical gumbo, and the brain is a computer. If you take those two different approaches, no wonder you're not going to kind of meet up in the middle somewhere.
We have time for one last question. If you are or wanted to be in the queue, but you couldn't ask a question due to the time constraints, we will invite you to carry the discussion later in person.
Thank you very much for this excellent talk and for guiding us through the concept of scientific revolution. I wanted to ask you about what do you think about how this evolution and revolution, let's say in neurobiology, follows the Karl Popper's theory of falsibility? Because we have a theory which was very strong that the neurons do not evolve, and we have a counter theory that new neurons and counter theory, new neurons.
Yeah, good. No, I think that's an excellent question. I think this case certainly fits Popper's bold conjectures and ruthless refutations model better than it fits Kuhn's. But nevertheless, remember, Popper's approach is still heavily theory-driven. The conjecture is a theory. Remember, think of his dynamics for science. You offer a conjecture, and then you try your darndest to falsify your conjecture. Now that is still an approach that is where theory is doing the guiding, and my picture is that when we really dig into the historical details of this case, not just what gets published in the scientific publications, because we all know those are highly refined pieces of writing, but also looking into the secondary literature, what the scientists say about their discoveries, what writers that talk about the scientists say about what the science. So if you take that whole, and then when you get a chance, lab notebooks, grant proposals, which we can now, successful grant proposals, we can get them through a Freedom of Information Act in the USA. You can look at this kind of whole panoply of evidence. And what I can't claim you find is that the tools drive the research, not the theory. And Popper's model has got theory front and center because that's what you're trying to refute. And I contend that when you really dig into the detailed history of these cases, using all the resources you can find, that's not the kind of structure that you find in these cases. But excellent point.
But then I would maybe argue that this could also be a Hegelian or maybe Marxist approach about how the means and the necessities of production, provide the tools for this.
Yeah. No. Look. We can talk about this. That's going to. I mean, I don't know. I don't know whether or not that's going to be the right account or not. I'd be willing to hear it and sort of play it up against the full set of data that I can find about any given tool. But I really contend that, look, the motivating problem for a new tool can be highly theory dependent. Because that's why you're looking for a new tool, because the old tools are just not simply providing you what you're trying to do. So that's going to clearly, I mean, you know, why get rid of that 14 carbon radioactive label? Well, because it's got too long a beta particle wave to emission in order to find something, to locate something as small as a chromosome. So that's highly theory dependent for that motivating problem. But I contend that when you actually try to build that tool, it's just messing around, just trying to find something that will do that particular job. And I think I've got some really good evidence from the way David Hubel built the metal microelectrode back in the late 50s that then revolutionized neurobiology for 25 years, and some really good evidence that a similar kind of tinkering was involved in Naher and Sackman building the patch clamp, which basically, as its importance, it rolled in the whole molecular wave into neuroscience. But excellent point.
Okay, so that wraps up our Q&A. So now I think we can have still a 15-minute break, just to get some fresh air in this room, and then we will start another talk, and we will tackle the topic of virtual brains. So stay with us. And let's thank again Professor Bickle for...
Thank you all.